Hi everyone. This video is an introduction to your red cabbage and pH lab. So go ahead and pull up the procedure, make sure you read it all the way through, and let's get started. I am looking at part A, and it's talking about anthocyanins. So it says red cabbage is an interesting plant. It's also known as a purple cabbage, a red kraut, a blue kraut, because it actually changes color depending on the pH of the soil. If the soil is acidic, the leaves are more of a red color. If it's neutral, more of a purple color. And if you have an alkaline or a basic soil, then you get more of a green or yellow color. This is due to a special pigment found in red cabbage called anthocyanin. Okay, this is really neat. If anyone has hydrangeas in their yard or have seen hydrangeas before, those are those big clumps of lots of little flowers, and they can either be pink or blue or white or maybe purple. That's the same type of idea. So those flowers actually change colors based upon the soil that they're growing in. And so the red cabbage you buy at the store or the purple cabbage is probably going to be a deep purple color. And that's because it's grown in more of a neutral soil. However, if you were to take that same plant and change the soil acidity, then it would change the color of the cabbage itself. So pretty neat. So for this lab, hopefully you went out and you bought yourself a nice red or purple cabbage. You can find them at Kroger for a few dollars. And that's going to be very important for this lab because it will be our indicator. So helping us determine if something is acidic or basic. Okay. Looking at part A, number 1, it says research what anthocyanins are and write down a few notes. Include a definition of pigments and how anthocyanins and pH are related. So I'll let you guys do that on your own. As a reminder, always make sure that you're doing good academic research, meaning you're not just using Wikipedia, but you're using .edu sites. .org sites can be pretty good too. Or you can even use the library. They have a good website with a bunch of articles as well. And then we're going to get into part B. So part B is the actual experimentation today. We're going to jump right into it. And we are going to make a pH indicator out of our cabbage. Now, some of you might have seen pH indicator strips. Maybe if you ever were a lifeguard or you took care of a swimming pool, you can actually get pieces of paper that act in the same way. They have some chemicals on them, and so when you dip them in different substances, the color of the paper will change, telling you if it's acidic or basic. We also have something called a universal indicator that we use in the lab, so in person, but that chemical is rather hazardous, and so we don't ship it to anyone's houses. That'd be a bit too dangerous. So you might see something like that in the future in labs, but in your house, a red cabbage offers almost the same thing. It's a pretty good substitution for it. So this is a pretty neat way to do it in the safety of our homes, our apartments, wherever we might be performing our lab today. So reading through part B, it says red cabbages offer a great alternative to using universal indicator, again, that more hazardous chemical, because it's very effective at finding pH. Well, so is universal indicator. They both are. But again, that indicator can be a little bit hazardous. So once you have your red cabbage, take a look at the rest of the materials and make sure to get everything all assembled before you continue. So you might want to pause the video and go through and find all of your equipment. Out of your lab kits, you're going to want to get one of your beakers. You're going to also want to get your goggles, of course. Okay. You'll need some glass containers. These can be actual drinking glasses. Okay, I'll walk you through what to put in them and how to clean them. Or maybe you have some plastic clear cups that you're going to use, but clear is going to be best. So you'll want some containers. You want some type of paper label. So either you want Post-it notes or paper and tape because we're going to be labeling the different cups. You also are going to have a big list of things that you might need. To add to this list, you're going to need a knife and a cutting board because we're going to be cutting up the cabbage. You're going to need a pot so that you can boil it with some water. And you might also want a strainer. Okay, so a different colander or a salad spinner, something like that if you have it available. Or even just a slotted spoon can be used for that purpose. Okay. Now, all of these items, it says possible items to test under the materials on your procedure, and you do not need to use these exact ones. These are just some ideas. But what you need are 12 different items that you can test the pH of. Now, they don't have to be a liquid already. If they're not a liquid, I'll show you how to make it into a liquid. Okay, if it's something like baking powder, you're going to mix it in some water. If it is a liquid, maybe it's something that you need to dilute, like liquid soap. We don't want to use a ton of your hand soap. We'll just use a little bit and dilute it with water. Okay, so you're going to be using your tap water for a lot of this. It might be helpful to be near a sink. You could test bleach. If you do so, remember that's a very strong chemical, so please handle it carefully. You might want to look up the SDS sheets so that you know what happens if you accidentally get it on your hands. Okay, you're going to be wearing goggles, of course, so you shouldn't get it in your eyes, but please be careful. Maybe you want to test some different drinks. So maybe you test out Sprite or orange juice or milk. Maybe you test wine. Okay, maybe you test vinegar, lemon juice, basic water. Maybe you test some shaving cream. You could try your shampoo, your conditioner, maybe some sugar, maybe an antacid like Tums or Maalox, maybe cream of tartar. So a whole bunch of different things that you can try to test out. These are, again, just some ideas, but you don't have to use these exactly. You need to find 12 different household items. Use some where you know what the pH is, right? Lemon juice, we know that's acidic, right? So use a few things as your control. You know that lemon juice is going to be acidic, so that will help you see how the Cabbage indicator will react to tell you that it's an acid. That's more like your control for your lab. But also you want to test out things that you don't know, because that's part of the fun of experimentation. So I'm going to pause the video and I'm going to set up my lab. I suggest you do the same. Make sure you have all of your safety procedures around. You've told your family members that you're conducting a lab. You will be using a stove and you'll be using glass containers. So please make sure that everything is safe for those around you and safe for yourself as well. Okay, I am now in my lab, so hopefully you are as well. I have my red cabbage. Here it is. I have some glass containers to do our testing. Now, ideally, you would have 12 of these containers. I don't have that many glasses, so I'll show you how I'm going to do it, just in case you don't have 12 glasses either. I have a beaker from our lab kit. And I also have this lovely measuring tube here. Remember that you have a base with it in your lab kit as well, and it will just pop straight down so that you can stand it up on the table. So you'll need this, you'll need your glasses, you'll need a knife and a cutting board to cut up your red cabbage, and then you might also either want a colander or some kind of slotted spoon, salad spinner maybe could be used, and you'll need a pot for boiling. Now you'll also need all of your other testing items. So you want to go through and make sure that you have 12 different items ready to go to test and some labels for them as well. Now, first things first, your lab says that you're going to actually cut up your cabbage into about 1 centimeter strips. So again, you can use your pinky. About the width of it is roughly 1 centimeter. Okay, so you want to go through and cut your cabbage up into little strips. Now you can leave it as a strip, or what I did is I went through and I diced it up into even smaller pieces. You want to cut up about a half of your head of cabbage. So you should have a nice pile after you're done. Again, make sure to cut carefully, wash your knives afterwards, just like if you were cooking in the kitchen. And again, you want some thin strips of cabbage. If you dice them up smaller, that's perfectly fine, but make sure they're at least a 1 centimeter strip. Now, once you do that, you're going to put this inside of your pot and you're just barely going to cover it with water. You don't want to put in too much water because then it would be too dilute. Thinking about our molarity, right? We'd end up having a lower molarity solution. So make sure that you just cover it with water. And make sure you have a large enough pot to do so. So I now have my cabbage in my pot. I'm going to add a little bit of water just to cover the very surface of it. And you're actually going to boil this and let it sit for 20 minutes. So you want to get it to boil and then you just want to let it sit. So that way it starts to extract and pull out those anthocyanins or those pigmentations that makes your cabbage purple and also helps us test the pH. So I'm going to go ahead and cook mine and I'm going to let it sit and then we'll be back. Okay, So now you've boiled your cabbage, you took it off the heat and you let it sit for 20 minutes. So now we're gonna go ahead and strain out the solid parts, the actual cabbage leaves. You can dispose of them. All we need for the lab is the liquid that's left over, and it should be a really rich, dark purple color. So you can either use a slotted spoon to pull out your cabbage, or you can use a colander just to dump it in. But if you do the colander method, make sure you need something underneath to actually catch the liquid. So go ahead and strain out your solid. Make sure you let your liquid drip a little bit, and then again, you can just dispose of it. And once you do that, you're going to be left with your pH indicator. Okay, so now I have strained out my cabbage. I have a nice, rich, deep purple color. Kind of hard to show without spilling it, but it should be a nice, pretty color. So I still have a little bit of solid in there, but most of it's strained out, and that's okay. A little bit of solid that's left won't affect your results. So at this point, you want to make sure that you have your goggles, put them on. This liquid could stain, so if you want to wear an apron or an older t-shirt, that might be a good idea as well. And you might also use a funnel if you have one in your kitchen that could help you pour this liquid out. I'm going to use a ladle just to kind of ladle it out and do it that way, so whatever works best for you. Now you should have your 12 different items that you want to test, and ideally you have 12 different glass containers to put them in. Now again, if you're like me, you don't have 12 glasses that you can use for this. So what you're going to do instead is you're just going to make sure to rinse each one out thoroughly. Use as many as you have, and then be sure to rinse them out. Now we want you to have as many glasses as possible because it's easier to see the pH if you line them up. And then you can see the color gradients as they change from acidic to basic. So if you don't have 12 glasses, it's okay, but get as many as you have and try to keep everything lined up so you can see the differences in the color. Now, there's a few notes on your procedure. It says you're going to fill up the container with 200 milliliters of substance, so that's where your beaker comes in, that came with your lab kit. Now, whatever substance you use, remember if it's a powder, you can actually, if it's not a powder, you can grind it up, like if you use an antacid tablet. But if it's in some kind of solid form, get it to a powder, and then you're going to have to mix it with neutral water, right? Just some regular tap water should be fine. The more of that powder you add, the stronger it's going to be, the higher the molarity, the stronger the concentration. So your procedure doesn't tell you exactly how much to add. That's for you to experiment with. Maybe just a couple teaspoons will be fine, but you can play around with it and see if you add more, if that changes the color of your indicator. So for example, if you have baking powder you want to test out, you're going to put baking powder with water to the 200 milliliter mark on your beaker. So you want to have a little bit of baking powder, maybe a few scoops, and then fill it up with water to the 200 milliliter mark. Give it a nice swirl, mix it all up, make sure it gets as mixed as it can be. And then you're going to add your indicator and see what happens. If you don't see much change, maybe you add a little bit more of your solute or your stuff to it and see if that does anything. Now, if you're using very strong acids or very strong bases like bleach, please be careful with it. You only want to add a little splash. And it's always best if you make sure that you're pouring very slowly so as not to splash any of the chemical up in your face. Okay? We always say it's better to add the water first and then pour a little bit of the bleach on top so that you don't actually splash bleach when you're adding all of the water from the sink. But either way, however you do it, please be careful. And if you are using bleach or some other strong acid or base, use your common sense, right? So I'm using my kitchen drinking glasses. If you are as well, don't put bleach in them, okay? Only put things in there that you can wash out thoroughly and you know are not going to be toxic to you if you later drink out of these. That is a bit of the caveat with our at-home labs. We can't keep everything as isolated in the lab as we would like to. So if you want to use a bleach or something like that, please make sure it is a disposable container in whatever form that might be. If it happens to be a colored cup, that's okay. Just make sure that you can see the inside, that the inside is white, all right? So again, use caution as you do all of this. Now what we're going to do next is I'm going to start testing out some things. So I actually have this rather interesting contraption. It's a water purifier, but it can actually turn my water into either an acidic water or a basic water. And I've had it in my house for a long time. I use it for a lot of different things, but I've never actually tested if the water is acidic or basic, like it's supposed to be. So that's what I'm going to start with. I'm going to start with some acidic water out of this purifier and some basic water out of the purifier. Now, if you just have regular sink tap water, it should be neutral, okay? But I'm going to go ahead and try out those two different things. And so when you try things out, you want to make sure that you have your cup labeled with a Post-it note with some tape. Just make sure it stays on there and you can tell what everything is, okay? I'm going to be testing out cashew milk, some coconut milk, some yeast. That's going to be my powder. I'm going to mix in some water. I'm going to test out my acidic and my basic water. And then I also want to test out black tea. So I'm going to make some black tea and see what happens with that. Now, you'll have another 6 to do, so you want to have 12 total. And if you're reusing containers, just make sure to give them a nice thorough rinse in between. Now, your dish soap could have a certain pH to it. Maybe that's something that you test. So if you go, just make sure to rinse it thoroughly with water. And again, as you go through the lab, we want to think about being safe. So when you do dispose of these different chemicals, make sure you're doing so in a safe way. And what I mean by that is, say you use bleach, something that's very strong, you want to make sure that when you pour it down the sink, you rinse it with a lot of water to help dilute it as it goes through your drains, so it doesn't do any damage to the drain, to the sewage pipelines, to the waterways. So if you have strong acids or strong bases, the best way to get rid of them is going to be to rinse them down your sink with a whole lot of water. Now, we shouldn't be using anything more dangerous than that, okay? Certainly there are proper ways to dispose of chemicals in a lab. You can read about those in the SDS sheet. But for the things you have in your home, and most homes, they should be fairly safe to put down the drain with a lot of water. All right. I'm going to go ahead and fill up my different cups. And the way I'm going to do that, again, is by getting 200 milliliters of the liquid. So I want 200 milliliters of all the different things that I'm testing. And I'm going to start with let's start with my acidic water. So I'm going to pause the video. I'm going to go over to my purifier and I'm going to fill this up to my 200 milliliter mark and add it to my sample cup. Okay, I have my acidic water out of my machine. I'm going to put it in the acidic cup. There we go. And now I'm going to use this little test tube to measure out my indicator. Now, if you look at your procedure, you want 200 milliliters of whatever you're testing to go into your cup, and then you want 25 milliliters of your indicator. Now, you might want a funnel for this. If you don't have one, maybe you use a spoon or a ladle. But however you do it, be very careful, again, not to spill, it might stain. And you want to get this up to the 25 milliliter line. And your liquid might still be a little hot, so just use caution. I went a little over, I'm going to pour a bit out. Okay, now you have a nice graphic on how to eyeball exactly the 25 mark. And if you look at your procedure, it says that what you want to do is look for the meniscus. So the meniscus is a curve of liquid. It usually happens with water and other more thicker viscous things, especially because water has all those neat tendencies we talked about. And so when you see a curve in any kind of liquid that you're measuring, you actually measure it from the bottom of the curve, okay? Not the top portions, but the bottom. So I'm going to look straight on exactly at it and make sure that I am right at my 25 milliliter mark. I need to get just a tiny bit out. And there we go. So you're eyeballing to get exactly to that 25 milliliter mark. And if you have that meniscus, you take the measurement from the bottom. So now I have this weighed out, or measured out, I should say. I have my sample in its cup ready to go. And I want to make a prediction or a hypothesis on what's going to happen. So do I think that my acidic water is acidic or basic? Well, I'm hoping it's acidic, right? That's what it should be. We'll see if that's actually correct. So I'm going to go ahead and scroll down to number 4 on my procedure, and you'll see a nice table. You're going to fill in this table as you test out all of your different items. So it's going to ask you what the household chemical is. For this, it's acidic water. You want to predict the pH. So I'm going to guess that this is acidic. And then you'll write down what the actual pH is. Now, how do you know, right? All we have is this purple liquid. How do you know if it's acidic or basic with cabbage water? If you keep scrolling down in your procedure, you'll see a nice table called the red cabbage indicator color chart. These different colors will give you a rough estimate on if it's acidic or basic and what the pH would be. Now, it's kind of qualitative, right? We're assigning a number to make it quantitative, but those colors are subjective. So in a lab, this might not be the most accurate way to do it, but color is a very common way to test pH. And so having that difference between the red on the very acidic side and the green-yellow on the very basic side can give us a good range in the color spectrum to see what's actually happening. So knowing that I'm starting out with a very deep purple color, I can see that's very close to the 7 on this cabbage indicator scale. And so sure enough, I'm just going to go ahead and add this to it. You might need a spoon to stir things around. Again, if it's a spoon that you're going to eat with later, please make sure that you wash it and you don't put anything dangerous with it. But you're going to go ahead and add, and then you want to investigate what this color looks like. Now, it might be helpful to hold a sheet of white paper behind it so that you can really see. Yeah, this is definitely lightened up. It got a little bit more to the purple-pink side of things as opposed to that really deep purple color that it started out with. Purple-blue color, I should say. And so I'd say, sure enough, this is acidic. Now I want to keep this cup around because, again, we want to spread them out so we can see the whole range of things that we're testing as best as we can. So I have to use the same beaker for all of my different samples, which means I'm going to be rinsing this out very thoroughly between everything that I do. So I'm going to go ahead and I'm going to fill up my basic water now, and let's see what happens with that. Okay, I now have my 200 milliliters of my basic water that I'm going to be testing. I'm going to put it in my basic water sample cup. And then I have 25 milliliters of the indicator. So I'm going to go ahead and pour that in, and we'll see what happens. Oh, it's kind of fun. So my basic water should have been basic. I'm going to hold it up to something white so that I can see. And this in person is actually quite a very pretty emerald green. So it looks a little bit different in my computer's camera, sorry. But it's a really pretty emerald green. So I would say that this definitely is basic, like we were expecting. And it looks pretty close to that 10 mark of pH. Nice pretty color. Side by side, they're certainly quite different. Okay, so I'll keep those there and we'll keep testing. I'm going to make sure to rinse my beaker out again between everything that I do. Now the next ones I'm going to get started are cashew milk and coconut milk. I've never tested the pH of those, so why not? And then I'm going to do some tea and some yeast. Now the yeast I have is actually a powder. It's called nutritional yeast, and I like to eat it on a lot of things. And so I'm going to take this powder, and as I mentioned before, if you do use a powder, you're going to add some to your beaker, a few scoops maybe. We don't know how much, right? That's part of the experimentation to see what happens. You shouldn't need a ton, but maybe a few teaspoons, and then you're going to fill it up to the 200 milliliter mark. If you're doing something like tea or coffee, maybe you want to use some hot or not boiling hot, but some warm water to help dissolve it. If you're testing sugar, that's a good way to dissolve your sugar as well. So I'm going to go ahead and get those things ready, and then we'll line everything up and see what happens. Okay, I now have my different samples lined up. I've already tested my acidic water, and it was in fact acidic. And I tested my basic water, and we got this nice pretty basic color green. And so next I'm going to go ahead and do my cashew milk, my coconut milk, my black tea, and my yeast. Now my black tea and my yeast were powders, and so I made sure to add some tap water, which we consider neutral, to make sure that we can get a liquid to test it out in. Now I have my 25 milliliters of my indicator, and I'm going to go ahead and add it to each one of these and see what happens. So this first one is my cashew milk. It's a nice pretty color. Now remember, as you're doing this, you should be making predictions about what will happen, if it will be acidic, basic, or neutral. This is my coconut milk. Okay, that's pretty close in color. I'd say my coconut milk definitely turned more blue though. So that's probably a bit more basic than my cashew milk, which maintained mostly that nice lavender color. Now remembering that it actually started out as a white color, it's going to be a little bit different than your clear liquids. And my next one I'm going to do might be a bit tough to tell the color change. We'll see. So my black tea is a bit of a dark color, right? It's pretty concentrated. Maybe you test out tea and you do a little bit less, or you do a green tea, or maybe you try more diluted coffee. So we'll go ahead and see if we get a color change with this, and if we don't, maybe I just use a small amount of that and put water in it so it's more dilute and we see a color change. Let's give it a go. It actually does look a little bit more pink. We're going to let some of the sediment settle and get a more accurate answer here in just a minute. And my last one is nutritional yeast. So again, I want 25 milliliters, eyeballing it at your meniscus level. Okay. And remember, my yeast is starting out a bit yellow, so that will change. And there we go. Ooh. Okay. Now, if you have something with sediment, it might be worth letting that set a little bit so you can see really clearly the liquid involved. And I'd certainly say that turned more of a green color for my yeast. Okay, now it started out yellow, which means it's probably a blue. So that's going to be a little bit of the issue when you start testing out things with colors. So clear liquids will give you the easiest answer, or white liquids as well. So you can see a little bit of the confusion or the difficulty if you test out liquids of different colors. It doesn't mean don't do it, but you might need to take in the color change from going to a yellow into a green. Probably means I added a blue to it, right? So this one, basic, but I can't really tell how basic. My tea, in person, it's actually looking decently pink. But again, it started out as a nice deep brown, so it's a little bit hard to tell exactly how acidic that tea is. Now my coconut milk started out white, so you guys can probably see that's a nice pretty sky blue. I'd say that's definitely basic. And then my cashew milk is probably more of a neutral color. So it's actually a nice light purple or a lavender. And that started out as a white beige kind of color. So go ahead and try it out with your samples. Now these are my 6 different cups. You can line them up. This was my basic water, right? My acidic water, okay. Try out a few different things. Maybe you try out some lemon juice and get a nice pretty pink, right? We would think that should be acidic. But maybe you want to compare the acidity of lemon juice and orange juice, maybe even to apple juice, okay? So try out a few different things. And if you get some murky results, that's okay. Test it out again. Maybe you take a little bit of this and you dilute it with more water so the color is more clear and easier to see when you add your indicator to it. So as always, have fun with it. Grab things that you don't know what they're going to turn into and test it out. And once you're finished, make sure to take pictures so you can include them in your lab analysis for your homework before you throw anything away. Now, once you are finished, you've gotten all your pictures, you've tested everything out, remember to dispose of these in a proper and safe way and clean out your containers, especially if they're containers that you use for drinking or eating out of. Okay, so now you've finished your lab, and make sure again to take pictures of your samples before you dispose of them properly. And also a reminder, do not throw away your beaker or your test cylinder. You want to make sure to rinse those out thoroughly, let them dry, and then keep them with your lab kit in case we need them for future experiments. So after you've cleaned everything up, you're going to want to go through and we'll look at number 5. It says rewrite your items in order from the lowest pH to the highest pH. So the most acidic or the closest to red, all the way up to the closest to that green-yellow high basic color. Now, depending on what you tested, you might not have the entire range. That's okay. But put them in the best order that you can, judging the pH by the color change, as we talked about. And if you get some murky results like I did with the tea, again, maybe you have to retest. You should have enough of that indicator solution from your cabbage so that you can test it out a few different times and line them up and try to get an idea of what the basic things were, what the more acidic things were that you tested. You have a place to insert your picture for number 6, and then a reminder to dispose of everything properly. Now we're going to look at the analysis together. So the analysis says that pH is a measure of the concentration of hydrogen ions in a solution. That might be a good thing to know for a test. So pH stands for the power of hydrogen, and we're talking specifically about those hydrogen ions, H+. So pH is a measure of how much H+ is in a solution. The more H+ there is, the stronger the acid. Now there's also something called pOH, which means that the OH- are the basic things. So something with a lot of OH- is going to be very basic. Now we don't talk about pOH. You might hear that in the future, and it's just a different scale to measure the OH. But in this case, for pH, if something has a lot of H+, it's very acidic, and if it has a very low amount of H+, it's going to be basic. Now on our scale, pH goes from 0 to 14. 14 is being very basic, 0 is incredibly acidic. 7 is in the middle and is neutral, not acidic or basic. So it says strong acids have a low pH, strong bases have a high pH. Weak acids and bases are closer to 7, the pH of water, which is neutral. Now, number 1, acids have more blank. Number 2, bases have more blank. They want to know the ions. So what ions? Well, acids have more H+, right? And what did I say bases have more of? Fill that in. And number 3, just some reflection on your experiment. So what items did you test that surprised you? I actually had no idea how my cashew milk was going to turn out. Okay, so that's kind of interesting. Did you make accurate predictions? Go back through, think of what your hypotheses were for your different samples. Were you correct or not? Make some general statements about what kinds of things we use acids and bases for in our homes. So of the things you tested, the stronger acids and the stronger bases, what were they and what are their purposes? Do we do certain things with acids or certain things with bases? What do you think? And then it says research general descriptions about acids. Write them here. Now that's a very open-ended question. I'd maybe have at least 3 different bullet points about acids. Okay. Now again, you want to make sure to be researching on reputable sources, .edu.org, or actual peer-reviewed scientific articles. Okay, so make sure you're finding good sources, but research a little bit about what acids are, what their purpose is in the world, how we make acids maybe. And then you're going to do the same thing for bases. Now it could be anything that you're curious about also. Again, we want to help you think of all these questions and stimulate curiosity. So if there's something about a base that you really want to know, go ahead and research it and write what you find here. Number 4, it says an element is any material that cannot be broken down into simpler materials by a chemical process. So it's at its base, it's the atom that we see on the periodic table. A compound, on the other hand, is 2 or more different elements chemically bonded together. So what 2 elements make up water? Number 5, give a few examples, at least 3. You should be able to fill this whole table up though. You want to give a few examples of some elements and the compounds that they make. So think of a compound. We've talked a lot about glucose. You know the chemical formula for glucose from photosynthesis and cellular respiration equations. So what elements is glucose composed of? Think of things in that way. What kind of compound do you have? You can even research this if you want. Maybe you're curious what bleach is made out of, and so you research the chemical formula for bleach, right? Write down the compound, write down the elements. Give at least 3, but you should be able to fill out quite a few more. And that's the end of your analysis. Now, I hope you review this video to help you study for the test because we will be talking about pH on a few of the questions. And so knowing that pH is a measurement of the hydrogen ions will be very important. You also want to know that the scale goes from 0 to 14. And as we mentioned during the lecture on pH, as you go from one number to the next, one whole number to the next on the pH scale, you're actually going up a range of 10, okay? So something that is a pH of 9 is 10 times more basic than something with a pH of 8. So it's scales of 10. Every time we move up a whole number or down a whole number, we're actually going a fold of 10. So something with a pH of 2 is 10 times more acidic than a pH of 3. All right? Now remember, 7 is going to be neutral. And there's one other concept for you to think about. And we didn't test it out today. It's kind of hard to get the supplies, but your body does it all the time. And so this concept is something called buffering. So a buffer is a salt, a substance, a compound that can help balance the pH and keep it from dropping too much or raising too much. It keeps it more in a homeostasis, more in a healthier range. What I mean by that is, think of our bodies, right? If we ate a lot of things with lemon juice and orange juice and apple juice and tomatoes and coffee, we eat a lot of acidic things, right? My body's pH is between 7.35 and 7.45, or else I start feeling pretty sick. So if I'm eating all of this acidic food, shouldn't my body pH then be lowered? Shouldn't I become more acidic? And certainly that can happen to an extent, but your body does something called buffering, and that it's able to help neutralize when you add acids or bases so that it doesn't go too far out of range one way or the other. So a buffer is a way to keep the pH more stable, okay? It doesn't mean that it won't change at all, but if I have a buffer in a solution and I start adding acid, I'll have to add a lot of acid before the pH drops. Whereas if I have the exact same solution without a buffer, I could just add a few drops of acid and that pH would drop right away. So the buffer helps to balance, either going from an acid into a more acidic thing or the base getting more basic. That buffer is incredibly important, again, in the human body so that our pH doesn't get outside of that ideal homeostatic range. We won't go into a lot more detail on how that all works. Your book does have an example about the bicarbonate system in the body you might want to read about, but just understanding roughly what a buffer is and how it relates to pH will be important as well. So I hope you had fun with this lab. I think it's a fun one to do, and as always, feel free to email me or ask if you have any questions. Thank you.